Hi guys, so before we begin, let me ask you a very quick question. Would you prefer reading 5 pages of the resource like this and trying to remember everything as much as possible, or just going through three mind maps, just three concise mind maps, and remembering all the information that you need to answer any type of question in an exam? Well, that's what we are going to do today. As you can see, mutation is almost 5 pages of the resource and there's a lot of information. If you try to just decode this on your own, it's going to take a lot of time, and you might not end up knowing what's important when it comes to the key points here. So, I'm here to guide you and teach you all the parts that you need when it comes to mutations. This is somewhat confusing to some of you all because there's a lot of subsections that come under mutation, and I'll be covering all that today within a very short time period. So, let's begin without further ado. So as you can see, this will be one mind map that I told you all, and then one for gene mutation and another one for chromosome. This is all you have to remember. So let's begin. Firstly, what is a mutation? Mutation means like you can come up with this idea of if something is changed, basically something is changed. So a mutation means it's a permanent alteration in the nucleotide sequence. So we know nucleotide sequence, we have nucleotide bases and there are sequences in it, right? So if there is a permanent alteration in the nucleotide sequence, you call it what? A mutation. So, what are these mutations? They can lead to phenotypic variations, which means now when something is like it undergoes mutation, then you can see phenotypic. Okay, so if something is undergoing, so what is this mutation? What will it cause? So we know that if something is undergoing mutation, there can be phenotypic variations, which means you can see that something has gone differently. Something has happened to the organism for it to show some variations. And what is the significance of mutation? Now, just like anything in nature, this mutation also can be neutral, it can be beneficial, it can also be negative. Okay, so firstly, we have neutral mutation. Neutral mutation means there's no positive or negative impact from the mutation, just as it is. Then we have deleterious mutation. Deleterious means it's having a negative impact on the organism. So this can reduce the organism's genetic fitness, and also it can be lethal, which means the organism can even die because of this particular mutation. And we have another type that is beneficial mutation. So if a mutation is beneficial, it can enhance the function of a protein. So mainly we speak about changing the nucleotide sequence, right? So when we speak about nucleotide sequence, we know nucleotides are going to be encoding proteins, and that's how all these things happen. So now it can enhance the function of a protein, and also it can increase or create a new function. For example, we know enzymes are specific to a substrate because they have active sites and they're very specific to their substrate. So if there's a mutation, sometimes the specificity of an enzyme can also change because of a mutation. So we consider that as what? A beneficial mutation. And there's another one. Because of mutations, we can have evolution happening very rapidly. So, an organism's survival can increase, and an organism's reproductive success can also increase because of a mutation. So, now we know what can happen because of a mutation and what is the significance of a mutation. Now we need to know what are the types of mutation. Now this is like a broad way of categorizing mutation. Firstly, you categorize them based on the scale. So if this is a small scale mutation, small scale change, you call it as gene mutation. If it's a large scale change, you call it as what? A chromosome mutation. These are the two main like branches of mutation. So what is the small scale changes? Now, if there is an alteration in the nucleotide sequence within a gene, this can affect the protein, right? So, if the protein is affected, the phenotype can also change. So, if there are small scale changes, so what is the key term here? Small scale changes in the nucleotide. If there are small scale changes within the nucleotide, it's called what? A gene mutation. And what is a large scale mutation? If there is a change in the chromosome. So we know that genes are going to make chromosomes and so on. And then they're going to make proteins and all that. So here, if there's a change in the structure of or the number of chromosomes, it's going to be what? A large scale change. So this large scale change can result in what? The change in a genetic material or even the phenotype as a whole. So now this is the basic introduction to what's a mutation. So firstly, what's the significance? What can result because of a mutation? And the two main types of mutation. So now let's move on to the next slide. That's going to be gene mutation. So now I told you small scale changes are going to be called what? Gene mutation. So now we have to see what are the causes for gene mutation. What causes gene mutation? So firstly, it can be spontaneous, which means if there are some rare errors in DNA replication, that can also lead to gene mutation. Or we call mutagens. Mutagens means the factors that cause mutation, gene mutation specifically. So we can say chemical, they can be chemical, and they also can be physical. So examples are like for physical, you can say UV rays, X rays. So we know that when you get exposed to X ray, UV ray intensively, that can result in mutations. Okay. And next one, mutagens. Mutagens are carcinogens. So now you might have heard like you shouldn't take a lot of X-rays or you shouldn't expose yourself to some radiations because that can result in cancers. So carcinogens means factors that cause carcinoma. So that's factors that cause cancers. So this statement is very important because several times I have like checked in MCQs and all. This is a very common idea. Mutagens are carcinogens. So most of the time, this is like a very obvious statement. Then, now we know what are gene mutations, permanent alterations in the DNA sequence of a gene. Now, what are the types of gene mutation? So you can broadly divide this also into three main types. Firstly, you have point mutation, and then you have insertion, and then you have deletion. So what is a point mutation? Point mutation means if you change one nucleotide only. So let me show you something like this. If you have a sequence like this and you change only one nucleotide, then you call what? You call this how do you call this? You call this a point mutation. So what are the types of point mutation? So main type is going to be substitution. So substituting is going to be the main type of point mutation. So there are three main types. If you substitute something with another, it can either result in no change or it can result in something completely different or it can result in something really disastrous. Okay. So firstly, we have silent mutation. That means if you substitute a nucleotide with another, that can result in a protein same as the previous intended protein, which means now same amino acid. So now for example, silent mutation, even if you change one letter, okay, even if you change one letter, it's going to result in the same amino acid. Here you can see this T is being substituted with G, but still it's coding for the same amino acid. Now next one is going to be nonsense. Nonsense means if you are going to add a nucleotide instead of another, that is going to result in a stop codon. So we know UAA, UGA, UAG. Those are going to be the three stop codons that we know in our syllabus. And that's going to result in what? A stop codon. So right when there's a stop codon, what happens? It results in premature termination of the protein synthesis, which means the protein synthesis is going to abruptly stop. So what happens if the protein synthesis abruptly stops? That's going to result in a shorter protein or the protein could be non-functional also. So that's going to be for nonsense mutation. So nonsense mutation, as you can see here, instead of G, you substitute it with T. So then what happens? This results in a stop codon. Stop codon means what? The protein synthesis is going to abruptly stop. Next one is going to be missense mutation. So missense mutation, substitution, there's another type called missense. Now, what is missense mutation? Missense means if you substitute one nucleotide with another, that results in another protein, another amino acid. Sorry, not a protein, another amino acid. Now, for example, here, missense means now here you actually AGTC, that's going to code for an amino acid. But here what you're doing is you're substituting this G with A. So if you substitute this G with A, it's going to result in what? Another amino acid. That's going to be an example for what? Missense mutation. So there are three main types under substitution. Firstly, if you substitute something with another, there are three main possibilities. Don't try to remember it in terms of the names. Just try to understand the logic. So if you have a nucleotide and you substitute it with another, there are three possible changes. First, there won't be any change, just like a neutral mutation. There won't be any change. So it will result in what? A silent mutation. Or if you substitute with another nucleotide, then what happens? The amino acid itself changes, which means it will code for another amino acid. So what will happen? It will affect the protein's function, or sometimes it may not even affect. Then there's another type. If you substitute one nucleotide with another, then it results in what? A stop codon. So now for example, before the mutation, AUG is there. So the G wasn't there. Some other one nucleotide was there. Now right when you put AUG, there's a stop codon. Then what happens? Premature termination of protein synthesis occurs. Next, you have insertion and deletion. So now, even in insertion and deletion, now there's a small idea. It's not properly mentioned in the resource also. Now, insertion and deletion can also be point mutations. Why? If you insert one nucleotide or you delete one nucleotide, then you can consider it as what? Point mutation. But in the resource, they have considered it as what? A different subsection of gene mutation. Next one you have frame shift mutation. So now if you consider like that, first one is point mutation. That means only one nucleotide is changed. Either it's inserted or substituted or deleted. There's another one called the frame shift mutation. So what's a frame shift mutation? That's a shifting in the reading frame. So for example, now you insert a nucleotide, right? So that can result in what? Extensive missense mutation, which means it can completely code for a different amino acid, and that can result in non-functional proteins. But sometimes there are chances for it to not cause any severe changes. So for example, if it happens near the termination codon, it's not going to cause any severe change. Next one, you have possible new stop codons. Now, for example, if you do a mutation, that can result in a premature stop codon, just like what? Premature stop codon, we came across this before as well. Where? So, premature stop codon, you came across this here. Premature termination of protein synthesis under nonsense. So, it can even result in a nonsense mutation, this frame shift mutation. And there can be also shift in the reading frame, and then after the mutation happens, the reading frame can return to the normal one as before. So that's going to be an example where you have triplets. Now for example, you have triplets, so you insert three at once. If you insert three nucleotides at once or delete three at once, then what happens? It's not going to cause any severe issue. So, point mutation means if you change one nucleotide. Frame shift means usually if you change more than one. So, like two, three together, then sometimes it can result in like a means nonsense where you stop the protein synthesis. Sometimes it can result in a new codon, or sometimes it can really not result in any negative changes. So, under chromosome mutations, there are two types. Firstly, you can change the structure of the chromosome, or there can be change in the number of chromosomes. So what is the structure of chromosome changes? So it can be deletion, translocation, duplication, and inversion. So next one you have structure. So structure is going to be example. So we can say deletion. What is deletion? It's very obvious. If you delete some part of a chromosome, that's going to be deletion. So this can be lethal. Lethal in the sense it can even kill an animal, kill an organism. The next one you have translocation. So now what is translocation? You might have come across this term in plant also. Phloem translocation, which means you move something from one location to another. So here translocation means a segment is moved from one location to another. So when a segment is moved from one location to another, is there any loss of DNA? No, right? But here in deletion, is there a loss? Yes, there's a part of the DNA that's lost forever. But translocation, there is no loss of DNA, but it can result in what? The gene expression being altered. So gene expression can be altered due to what? A chromosome mutation. So let me show you the translocation. Here, these two parts are going to be switching. Here, they are going to switch the places. Whereas if it's a deletion, what happens? Now see, you have something like this originally. You take this part out. So now this is the new one. So that's going to be deletion, which means there is a loss of a DNA fragment or a segment. Next one you have duplication. Duplication means you're creating a duplicate of it, right? So there's an extra piece of DNA. So this will also not lead to any DNA loss, but it can have a negative impact on the phenotype and also obviously change the gene expression can be altered. So duplication means what? You create an extra copy and you're going to add it. So that's going to be duplication. And finally, you have inversion. Inversion means now, if there is a fragment, a portion of the chromosome, you're going to turn it upside down or change it in orientation. That's going to be what? Inversion. So here you can see originally it's like this. This part is going to be inverted upside down, and you're going to have it again like this. So there are mainly like four types. So here, insertion is not there in the resource. So insertion is almost like, where you insert a part. Okay. So you have inversion where you switch the sides, orientation is changed. Deletion means a part is lost. So DNA loss is going to happen. Duplication means you're creating a replica. Then insertion means you add a new part. And translocation means you move one DNA fragment from one place to another, one chromosome from one place to another. Next one, you have the change in the number of chromosomes. Now, this is also very commonly asked in MCQs where they have two main types of chromosomal changes when it comes to numbers. So, firstly, you have aneuploidy. What is aneuploidy? Aneuploidy means if you have one chromosome less or one chromosome more. For example, you have monosomy. Monosomy means you have one chromosome less than you should have. So your requirement is 2n, but you have 2n minus 1. And you have trisomy. So trisomy is very commonly, an example is going to be Down syndrome where you have trisomy. So that is when you have one chromosome more than required, more than the usual. So that's going to be an abnormal condition. Here also it's abnormal. Why? Because it has one chromosome less. So the reason for aneuploidy is going to be now what is basically happening is non-disjunction. Non-disjunction means now we know in meiosis 1, we know that homologous chromosomes have to separate and migrate to each pole. And in meiosis 2, the chromatids have to separate and migrate to each pole. So here, if either of these does not take place or if both don't take place properly, that can result in what? Non-disjunction. Non-disjunction can result in what? Aneuploidy. So, for example, in meiosis 1, if the chromosomes don't migrate to one pole, both poles, but both migrate to the same pole. Or in meiosis 2, if the chromatids don't separate, then they are going to result in what? Non-disjunction. Now we move on to the next type of the chromosome number mutation, that's going to be polyploidy. So if there are more than two complete sets of chromosomes, you call it what? Polyploid. So polyploidy means we can say triploid, hexaploid, tetraploid, octaploid, which means now normally we have two n, that's diploid, haploid, that concept. So if you have one, you call haploid. If you have two, you call diploid. Now if you have more than diploid, you call it what? Polyploid. So now in animals, you can say it's more common in invertebrates, but vertebrates also it's rarely found in examples like certain amphibians and certain fishes. And when it comes to plants, of course, you can find it more, commonly than in animals. For example, triploid bananas, hexaploid wheat, and octoploid strawberries. So this is very common. You can see in MCQs also constantly being questioned. So triploid bananas, hexaploid wheat, and octoploid strawberries. So this is all that you have to remember. I think it's better for you to now go through this because I have summarized everything that you need to remember under this topic into three mind maps, and this is all that you have to remember. So, if I'm going to like rush through the points again, you have mutations, you have to know the significance of it and what leads to that, and then the two types of mutation, and then you have gene mutation, which is a main subsection of mutation. What are the causes of it? And you have three main types: point, insertion, and deletion. And then you have to remember each subsection of substitution. And then frame shift mutation, what is that? What can result in that? And all that. And then chromosome mutation, which is another subsection of mutation. Again, you can divide it further into structure and number changes. And then what are the subsections and the causes of them and the examples. This is all you have to remember. So you can even summarize everything into one paper, and that's all you need to remember under mutations. So, uh, so go through this part again and see you in the next video. Thank you.